In this video, we're going to look at how prokaryotes divide their cells. So, since prokaryotes don't have a nucleus, their cell division is slightly easier and less complex than eukaryote cell division. And we call prokaryote cell division binary fission. And this is a type of cell division that occurs in prokaryotes. And this happens in 3 steps. The first step is the cell's chromosome is copied to make 2 identical copies. Then the 2 copies of the chromosome will separate from each other and move to opposite poles of the cell. So the one will move all the way to the left, the other will move all the way to the right. And so the poles would be like the ends of the cell as it's elongating. Then a cell wall will grow in the middle here, and the cytoplasm will split apart. And this is what we call cytokinesis, when a cell pinches into 2. So anytime in the next couple videos you hear or see the word that starts or has the part cyto in it, that simply means cell. So cytokinesis is telling you that it's doing something with the cell, and the word kinesis means to cut. So the cell is actually being cut into half or cut into 2. So here's your diagram of prokaryote cell division. We start up at the top, and that shows you the chromosome with the DNA in the middle. And then notice how that original DNA is that grayish purple color. In this step 2, you can see that the cell has started to elongate, so now we're more oval instead of circular. And you'll see that there's a new strand of DNA, that blue strand of DNA. So the DNA has been replicated. Then notice the DNA molecules are separating. They're moving to the opposite poles. And the cell wall right here is starting to form. Then eventually that wall completely forms, and we end up with 2 daughter cells. And these 2 daughter cells are identical to one another. They are same. And so that brings about pros and cons of binary fission. So pros are like the good things, and cons are the not so good things or the bad things. So one of the really good things about binary fission is that it happens very fast. It happens very, very quickly. Cons, the bad of binary fission is we get those identical daughter cells. So generally, when you have those identical daughter cells, if one of those daughter cells is going to be killed by an antibiotic or harmed in some other way, both of them will end up having the same problems because they're identical. And so being the same is not always good. And that's your prokaryote cell division.